For 40 years, the study of Alzheimer's disease has been trapped in an exercise of reverse causality. Researchers identify the terminal manifestations of the disease in the cortex, specifically amyloid plaques and tau tangles, and attempt to build a causal chain working backward. This approach ignores the spatial and temporal realities of the human brain. This chart visualizes a precise, unbiased stereological census of neuronal cell death, comparing populations in early-stage cognitive impairment versus late-stage disease. The arithmetic is severe. In the top row, the entorhinal layer 2 stellate cells, the primary gateway to the cortex, have already suffered a 60% absolute loss by a very mild clinical diagnosis. Yet this destruction is not a diffuse, uniform fog. On the bottom row, the cerebellum remains virtually completely intact, registering near-zero cell loss, even though it routinely accumulates diffuse amyloid deposits. If amyloid alone caused the death of the cell, the cerebellum would degrade alongside the cortex. Plaques and tangles are terminal byproducts of a highly specific 50-year structural collapse that begins far outside the cortex. To find the precipitating variables of neurodegeneration, we have to look at the massive, dynamic environmental interface that projects directly into the brainstem. We have to look at the gastrointestinal tract. In a healthy adult, the gut microbiome actively ferments dietary fiber into short-chain fatty acids. The most critical of these is butyrate. Butyrate powers the colonic epithelium and maintains the catalycidin checkpoint, producing an antimicrobial peptide capable of binding and suppressing amyloid fibrillation. As humans age, dietary shifts drive a reproducible ecological drift. Fiber-fermenting bacteria diminish, and pro-inflammatory taxa expand. The butyrate supply drops. The intestinal barrier fails, leaking microbial products into the blood, and the host loses its primary anti-amyloid checkpoint. This dysbiosis introduces a highly specific structural hazard. Expanding bacterial populations naturally secrete their own functional amyloids, like Curly, to build biofilms. These bacterial structures act as physical templates. Through a process known as cross-seeding, they make contact with enteric neurites and force human proteins to misfold into matching pathogenic shapes. This ecological shift transforms the gut into a chronic, highly active proteinopathic seeding compartment, generating a toxic signal that now requires a physical path to the brain. For an ecological failure in the colon to manifest as cognitive decline, a high-speed anatomical bridge must exist between the two systems. The interface begins with enteroendocrine neuropod cells embedded in the gut lining. These specialized cells directly sample the chemical contents of the intestine and translate that chemical data into rapid electrical signals. This diagram maps the exact neural route. The neuropod cells form fast glutamatergic synapses with the terminals of the vagus nerve. The signal travels directly up the vagus nerve, bypassing the blood-brain barrier, and enters the medulla's master control gate, the nucleus tractus solitarius, or NTS. The NTS does not route this data directly. It uses a dual relay system. The excitatory arm sends the signal to the PGI, which drives powerful, rapid-fire bursts upward to the locus coeruleus in the brainstem. Running in parallel is the inhibitory arm. The NTS projects to the PRH, which supplies a tonic, GABAergic inhibitory restraint, suppressing locus coeruleus firing and keeping the system in balance. A chronically inflamed, dysbiotic gut sends a relentless, noxious data stream through this exact circuit. The constant peripheral alarm heavily biases the excitatory arm. The PGI drive overwhelms the inhibitory restraint, trapping the locus coeruleus in a state of continuous excitatory overdrive. This unyielding electrical pressure from the gut strips the brainstem of its metabolic reserve. The disease has its physical foothold. The locus coeruleus is intrinsically vulnerable to this overdrive. Comprising only tens of thousands of neurons, it must sustain massive branching axonal projections to virtually the entire brain, firing continuously throughout waking life. It operates perpetually at the absolute ceiling of its mitochondrial capacity. Forced to handle the chronic stress signals from the gut, the resulting oxidative load fractures the neuron's DNA. The cell responds by hyperactivating PARP1, a DNA repair enzyme that consumes immense amounts of NAD+. This causes a catastrophic drain of the cell's primary metabolic currency, crippling its mitochondrial repair pathways. Instead of undergoing apoptosis and clearing out, these exhausted post-mitotic neurons enter a state called amyloid-senescence. They permanently exit normal physiological function, locking themselves into a state of suspended animation. A dying cell simply fades away, but a senescent cell becomes highly destructive. The amyloid-senescent locus ceruleus neuron begins broadcasting a senescence-associated secretory phenotype, a highly toxic, localized cocktail of pro-inflammatory cytokines and proteases. Those same vast axonal networks that once regulated the brain now become delivery mechanisms. They physically transport misfolded tau seeds and inflammatory secretions upward, dumping them directly into the forebrain and the hippocampus. Alzheimer's disease ignites with this clinically silent, localized brainstem senescence, a process beginning decades before the first sign of cortical memory loss. By the 6th and 7th decades, the pathological front driven by the brainstem arrives in the hippocampus. Here, the burden of the disease shifts entirely to the resident immune system. In a healthy cortex, microglia exist in a strict homeostatic state. They survey the tissue, support new synapses, and prune dead connections with exquisite restraint. As the locus ceruleus stops providing regulatory suppression and starts delivering toxic tau seeds, the local environment becomes hostile. Subjected to unrelenting oxidative stress, the microglia lose their homeostatic transcriptional signature. They age out of their custodial competence and enter microglial senescence. They accumulate lipids, become morphologically stunted, and exhaust their metabolic capacity, yet remain profoundly active through their own toxic secretions. Historically, researchers debated whether Alzheimer's was caused by microglia aggressively attacking healthy tissue or failing to clear amyloid debris. Cell biology reveals they are the exact same senescent state. A senescent microglion is clearance incompetent. It cannot clean up the tissue. Simultaneously, it constantly secretes inflammatory proteins, chemically burning the surrounding environment. The exact cell designed by evolution to protect the neural network has been converted into the primary structural driver of its degradation. This immune collapse targets a specific fragile architecture, the parvalbumin interneuron. These fast-spiking cells are the metronomes of the brain, synchronizing the high-frequency gamma rhythms required for working memory and cognition. Because they fire at extreme rates, parvalbumin interneurons rely entirely on the perineuronal net, a dense lattice of extracellular matrix wrapped directly around their cell bodies. This net acts as a rigid structural scaffold and a vital buffer against ionic overload. This diagram illustrates the proteolytic turn, the specific moment the senescent glia converge to demolish this protective matrix. It occurs through 3 simultaneous vectors. First, the senescent microglia release matrix metalloproteinases, which enzymatically cleave the protective sugar and protein chains of the net. Second, dying oligodendrocytes in the surrounding tissue release labile iron. This iron triggers Fenton chemistry, generating highly reactive hydroxyl radicals that physically shatter the structural core of the matrix. Finally, the classical complement cascade tags the exposed fragments with C1q and C4d proteins, marking the essential matrix for physical destruction and immune phagocytosis. Terminal dementia follows the physical stripping of this matrix. Without its shield, the parvalbumin interneuron faces catastrophic oxidative overload and shuts down, falling electrically silent. The rhythms of the brain collapse and cognition is severed. At this terminal stage, the localized synaptic destruction merges into a systemic failure. The entire brain is now locked in a self-amplifying loop. As senescent glia multiply across the cortex, they express high levels of the CD38 enzyme. This acts as a massive metabolic sink, broadly consuming the tissue's shared NAD+ energy supply. The localized bioenergetic burnout that started decades ago in the locus ceruleus is now mirrored on a massive organ-wide scale. Yet clinical data presents a profound paradox. Neuropathologists routinely autopsy the brains of individuals who harbored severe amyloid and tau burdens at the time of their death, but who exhibited absolutely no signs of dementia during their lives. These resilient individuals solve the Alzheimer's paradox. They remained cognitively intact because they successfully preserved the structural integrity of the perineuronal net. They maintained a population of healthy microglia and averted the proteolytic turn entirely. Cognitive survival relies on the maintenance of structural matrix architecture. When the physical scaffolding holds, the brain can tolerate the proteinopathy. This explains the repeated failures of single-target amyloid monotherapies. The pharmaceutical industry has spent decades attempting to stop a shifting, systemic, half-century network collapse with a static, late-stage tool. Interventions must match the temporal architecture of the disease. The highest leverage point exists upstream, in phase 1. By repairing gut dysbiosis and restoring butyrate production early in life, we can starve the vagocerulean conduit of the inflammatory fuel required to trigger brainstem failure. In later phases, the therapeutic focus must pivot to structural preservation. This means deploying rigidly timed senolytics to clear exhausted microglia, followed directly by protease and complement inhibitors to defend the perineuronal net from demolition. Alzheimer's disease is a process governed entirely by time. The window for intervention remains wide open, provided modern medicine finally aligns its pharmacology with the precise spatial architecture of the collapse.